Hi everyone. So the next series of videos is going to be going a little bit beyond one-way ANOVAs to discuss two-way ANOVAs or two-way analysis of variance. But first I want to go back to our diagram that we've been looking at when choosing a statistical test. We have our dependent variable, our y variable, which can be nominal or continuous, and we have our independent variable, our x variable, which can be nominal or continuous. And we're still in this section where we're looking at a nominal independent variable and a continuous dependent variable for an ANOVA. So we're still over in this far corner, but now we're going to expand upon the one-way ANOVA. So two-way ANOVAs are similar to one-way ANOVAs, but they do differ. And importantly, they can ask more interesting, complex questions than a one-way ANOVA. So in our one-way ANOVA, we had one independent variable. All right, and that's why we call it one-way ANOVA. Now this independent variable could have many levels. So we could have had populations A, B, and C. We could have had genders, male or female. We could have had drug. We could have had a placebo. Or we could have had actual drug administration. So there could be many levels of this one independent variable for a one-way ANOVA. In a two-way ANOVA, we go beyond this by having two independent variables. So two x variables. And again, we're not limited on the number of levels of each one of these variables. So say for example we have variable A and we have variable B. Well, we can have anywhere from 2 upwards of levels of these variables. So variable A could have 3 levels, and variable B could have 2 levels. So we're not limited on our number of levels aside from they have to be more than 2 levels. Okay, so let's talk a bit about two-way ANOVAs. Two-way ANOVAs look for interactive effects of our two independent variables. So the interactive effect of our two independent variables. So let's go with the example we've been using, which is mollusk shells and populations. And that was our one-way ANOVA, but let's add to that a little bit. So let's say that we still have our populations, which is our first independent variable. And in those populations, we have the Tillamook population, we have the Newport population, the Petersburg population, and the Portland population. Okay. So we have 4 levels of this one independent variable, right? These 4 levels. But now let's add a second independent variable to our analysis. Let's say, for example, salinity, water salinity. And this is going to be our second independent variable. And I'm just going to pick 2 random values. Let's say freshwater, so 0 salinity, and we'll say 100 parts per million. All right, so now we have a two-way design with our ANOVA. So let's talk a little bit about model effects. So as you saw in the SAS Jump program, there was a window for model effects. And what this is basically just organizing our variables into the analysis we want to do. So for a two-way ANOVA, our model effects would be as follows. We would have our first main effect, and that would be population. So that's our first independent variable. And I'm just going to say that that would equal P. And then we have a second main effect, and that is going to be salinity. And we'll have that equal S. So these are basically representations of each one of our individual variables. So if, for example, population, it would be the same thing, the same question we asked with our one-way ANOVA. Does the length of mollusk shells vary among populations? And similarly, for our second main effect, a one-way design would ask, does the length of mollusk shell differ between salinity treatments? But with a two-way ANOVA, what we're really interested in, the question of interest, is the interactive effect of these two main effects. So our last model effect would be our interactive effect, and it would be population by salinity. So population times salinity. So what does this really mean? What question are we asking with this interactive effect? Well, as I said, it's a bit more complicated. We're now asking, does the effect of salinity on mollusk shell length depend on mollusk population? So, does the effect of salinity on mollusk shell growth differ among populations? So with this question, we have posed whether or not there's an interaction of salinity and population. So salinity is one of our independent variables, population is another independent variable, and mollusk growth is our independent variable. So interactions can be a little confusing. What is an interaction? Well, an interaction basically implies that there are non-additive effects of our two independent variables on our dependent variable. In other words, you can add up the results of our two main effects have on y separately, and that would be, let me bring that back up. So if you add up the results that our two main effects, our two independent variables had on our dependent variable separately, we get the results of this main effect, but the interaction of those two variables leads to a result above and beyond the separate effects. Okay? So the interaction of the two variables leads to a result above and beyond the separate effects. So let me clear this off really quickly. Get a big eraser. Because I want to keep the figure up there so we can add in some observations. Excuse me. All right, so as I said before, you can have as many levels of each independent variable as you would like. So let's bring up our mollusk data again. You can see here that we still have population, our 4 populations. We have our shell length, but I've added salinity to this Excel sheet. And I've added it such that 5 of the Tillamook mollusks were subjected to fresh water, and 5 of the Tillamook mollusks were subjected to water that had a salinity of 100 parts per million. And I did the same for each of the other 3 populations. And this is how we would set it up in an Excel file with our populations listed and then our second dependent variable varying depending on the number of replicates we had. So in this example, we had 5 replicates per cell. So in other words, we had 5 Tillamook mollusks that were being subjected to fresh water and 5 Tillamook mollusks that were being subjected to saline water. And the same thing for each of the other populations, as you can see here. So that leaves us again with a total observation of 40 individuals, and our replication, our replicates, equals 5, because we have 5 individuals in each population subjected to each one of the salinity levels. Okay, and again, the question we would be asking here is, does the effect of salinity on shell growth depend on population? In other words, are mollusks from these different populations responding differently to the changing salinity levels? So in the next video, I'm going to show you how to enter in the data from a Twoway ANOVA setup into SAS Jump and how to run your tests that will answer this question.